So welcome. In this video, we'll talk about development, genes, and behavior. And so I'll switch over to my PowerPoint and get that rolling. Okay. So this material that we're covering is also covered in our other nature book, and that is in Chapter 3, Underlying Mysteries of Development. So we won't read all of Chapter 3, but quite a bit of it. Okay. We may come back to that later. And we're going to introduce the topic, take a look at what phenotypes means, focus on HOX genes, and then look at context and how context influences development. And so before we get started, I just wanted to introduce the ideas in this section by looking at this picture. This picture comes from our Mother Nature textbook, or our Mother Nature book, page 323. It's a picture of a Pakistani woman who gave birth to twins, and one was a boy and one's a girl. And she decided, along with her family, that she should nurse her boy and her mother-in-law could bottle feed her daughter. And you can see the outcome of this decision that she made. At 5 months old, her son was healthy and growing well, and her daughter died. And so genetically speaking, her little daughter may have been very healthy. We don't really know. But her genetic makeup had little impact on her outcome. Instead, it was the cultural environment in which she was born that influenced her development and influenced who she became. When we talk about development, we sometimes use the term maternal effects. Maternal effects include things like a mother's decision about how much energy to invest in her offspring. This is an example of a mother's decision, or maybe it was out of her hands. It's just one of the types of developmental factors, or one of the types of environmental factors that influence an organism's development and influence the organism's behavior. And in this video, or in this lecture, we're going to think about developmental contexts and how they can influence behavior. It's clear looking at this picture and thinking about how genetics and development have played a role in this little twins' growth and development and eventually death, that one's genetic makeup is inseparable from the context in which one lives, right? Genes are expressed within a particular environmental and developmental context. And under different situations, organisms with a fairly similar genetic makeup can follow very different pathways, different developmental pathways, if you will. We will call this phenotypic plasticity. One's phenotype can vary depending on the environment in which one is raised. And I think our textbook makes it clear and says it multiple times that one's genes are not inseparable from context. Genes are expressed within a context. And in this lecture, we're thinking about gene expression within a developmental context and thinking about how development can influence one's pathway, if you will. So before we get started, I wanted to make a couple of notes about phenotypes. This is a term that you probably don't hear that much unless you're a biology person, but a phenotype is the tangible properties of an organism. What one looks like, how one interacts, how one behaves. Those are, I have another definition here, I'm not sure I like it, but how one interfaces with the world and interacts with others in it. It's what you look like, who you are. It's not your genes, it's your expression of who you are. The expression, it's you, okay? Phenotypes are influenced by genes, but are not fully determined by genes, because phenotypes are produced by our genes and our interactions with the environment. And that could include our parents, our culture, whatever, okay? It is at the phenotypic level that natural selection acts. Natural selection acts on what you look like, are you able to survive that disease? How fast can you run? Can you see well enough to shoot an arrow? Those are the ways in which natural selection acts on phenotype. Okay, what are some environmental variables that could be important in producing phenotypes? What are some of the environmental variables that, that could influence one's genetics, if you will? All sorts of things. I have a whole list of them here. Diet, the time of year that an organism is born, viruses, prenatal exposure to different kinds of diseases or viruses, maternal effects, paternal effects, early learning, exposure to religious ideas, and on and on and on. There are so many environmental variables that could be important here. I have in my little notes, tiny little notes, that one thing that's interesting that I haven't mentioned yet is that an infant that is exposed prenatally to a virus could be have a very important influence on that infant 50 years down the road. Researchers have shown that individuals exposed to influenza in 1918, when they were in the womb, had a much higher risk of developing schizophrenia later in life, right? Wonder what being exposed prenatally to COVID-19 will do. We don't know. Okay, so when we're thinking about how genes and development interact, we're thinking about what happens at the phenotype level, all right? What happens at the level of the phenotype? What the organism looks like, how the organism behaves. Those are the phenotypes. Now, it turns out that genes and development are very tightly related, and they're related in one particular interesting way. There are genes that regulate development. These are called HOX genes, and HOX genes are described for us in our Agile gene, or the NVM is the same as the Agile gene book by Matt Ridley. But HOX genes are kind of clusters of genes or sets of genes that are conserved in different kinds of animals. We find clusters of these same kinds of genes. And what they do is they're expressed early during an embryo's development, and they contribute to the body plan of an organism. They help to determine what that organism is going to look like, what its phenotype is, but they are expressed really early in an embryonic development, and they have a huge impact on the organism's body plan. Here's a picture of some sort of fly, probably a fruit fly, but I don't know, and a mouse. And you see a cluster of these different colored genes on a bit of DNA here. And each of these genes is also found in the mouse, so from the fly to the mouse. And each of these genes contributes to the development of one part of this organism's body, the body plan, if you will. And you can see that the expression of each of these genes varies from fly to mouse, but they contribute to the same components of the fly and the mouse. And HOX genes are kind of interesting, and we need to really kind of think about how these genes work to understand them a little more. And this is a diagram that you don't have to fully understand, but I wanted to point it out to you just to think about how HOX genes actually work. Okay, so this is a picture of a bit of DNA. So there's our DNA along this length here. And on the DNA is a gene. And that gene is going to be, it's all in dark blue here. And that gene, I don't know what it codes for, okay? But that gene codes for the building of a protein, all right? Well, it turns out that all genes have what are called promoters, pieces of DNA usually kind of right next to the gene, kind of upstream of the gene, that help to regulate when this gene is turned on. When is it going to be transcribed and translated, made into a protein? And this is complex. It turns out that the gene is transcribed when a molecule called RNA polymerase, that's this thing, this little triangular shaped thing, binds to the promoter and starts to transcribe the gene, and that means that this gene is going to be expressed. It's going to start making its own protein. Okay? It'll be transcription and translation. But the kicker is, when does this RNA polymerase bind to the gene? When is it useful? Or when is it going to be able to transcribe the gene that it wants to transcribe? Well, it can only when transcription factors, like these guys here, bind to the promoter and draw in RNA polymerase. And RNA polymerase sees the promoter then and sees all these transcription factors and says, I have to bind there so that I can transcribe this gene, etc. Well, guess what? There's even more control elements on the DNA. We're not going to talk about those. But HOX genes are clusters of genes that actually produce transcription factors. They code for certain transcription factors, these little purple blobs up here, okay, that bind to genes and cause the gene to be transcribed. In other words, HOX genes help to regulate the expression of other genes. They turn on other genes. Their protein product does, at least. And that's really what we can say that they do. They code for the building of transcription factors, and those transcription factors are proteins that influence the expression of other genes. HOX genes contribute to kind of the regulation of many other genes, and they're typically active very early during embryonic development. And when they're active, they regulate or they turn on a whole suite of genes. And what's interesting is that in different animals, these HOX genes contribute to different developmental plans, because the HOX genes can vary in a variety of ways. Here's an example of one way in which those HOX genes can vary. They could vary in tiny little ways, small changes, mutations, if you will, or differences in these HOX genes, maybe even in the promoters of these HOX genes, can have an influence. So if we look at something like HOXC8 in mice and in chickens, these HOXC8 gene contributes to switching on other genes involved in the development of the vertebrae. In mice have 7 neck vertebrae and 13 thoracic vertebrae, kind of along the back, right? Chickens have 14 neck vertebrae, way more neck vertebrae, and only 7 thoracic vertebrae. They have the same HOXC8 gene, almost. Okay? Actually, the gene the HOXC8 gene promoter in chickens and in mice is almost but not exactly identical. Small changes in that gene contribute to differences in the expression of the gene and differences in the development then of the number of vertebrae in these animals. It's crazy. It results in a smaller number of thoracic vertebrae in the chicken than in the mouse. Okay? So tiny changes, mutations, if you will, in these genes can contribute to big changes in body plans. Researchers are also suggesting that the timing of HOX genes, when HOX genes are turned on, can have a huge role in regulating other genes and in determining the phenotype of animals. So when we look at dogs, for example, dogs have a very different phenotype than wolves, right? Dogs have, a different facial features. They have a shorter snout, a smaller skull, they have floppy ears. They also have very different behaviors, including a much more playful behavior in dogs. Researchers are suggesting that certain HOX genes contribute to the changes in morphology and in behavior that we're seeing in dogs compared to wolves, or that we've seen in dogs compared to wolves. And it has to do, we think, with the timing. When are these HOX genes active during embryonic development? How long are they active during embryonic development? Can contribute to a very different body plan and a different phenotype, behavioral phenotype, I should say, as well. So HOX genes are genes that contribute to development, early embryonic development, and that contributes to differences in body plan and differences in behavior that go along with it. And so here we have this connection between development and genetics. In humans, there's an example here that I wanted to cite. There's a possible link between several HOX genes and autism that researchers are studying. HOXA1 and HOXB1, especially HOXA1, has been studied, and they both seem to play a role in the building of the brainstem and the cerebellum. HOXA1 is actually active from 20 to 24 days after conception and plays a role in the development of the brainstem, cranial nerves, the ear, the skeleton, and the head. And our researchers have found that, and then if there's a problem with this gene, then that can lead to a problem in the development of all of these parts. What researchers have found is that there are some variations, some different forms of each of these genes. And for one particular version, 40% of people with that particular version of the HOXA1 gene go on to develop autism. That's about twice as many. Let me say that again. So 40% of people who have this one version of the HOXA1 gene also develop autism. That's a nice correlation. It's not a causation, but it is a correlation, especially because even there are other people who have this version of the gene who don't have autism, but it's much lower. The percentage is much lower. People who have this version of the gene are likely to have autism. They don't always have it, but they are much more likely to have it. It's interesting. Why? Because in some way, this different version of the HOXA1 gene, in some way, influence development that leads to the development of autism-like phenotypes. I don't know. It's something that's being studied. It's interesting. There's pretty weak evidence out there for it, but it is an interesting area of research because you've got this gene that plays a huge role in development, and that leads to our phenotype. Okay, we'll go to the next video in a minute. We'll come back.